grade 12s and welcome to Life Science. And today we're going to revise on response to the environment. Right, so we're going to look at the plant side and the animal side, right? So we're going to have a look at what we do, but mostly with the humans. Okay, so we're going to start off, if we have a look at the board here, we're going to have a start off and have a look at the key concepts. Okay, and the key concepts, we're having a look at this is what we need to do, or this is what happens in plants. Okay, and the first thing that we look at, the response that you have in plants, remember plants are only got to do with hormones. Okay, so we're going to look at specific hormones. The first hormone we're going to look at is called auxin. Okay, and auxin does specific jobs, and these are the jobs it does. First of all, does cell division. In other words, it makes the cell divide a lot quicker, make more and more cells. That's what cell division is. Okay, the next one, it does cell enlargement. In other words, it makes the cells bigger. If the cells go bigger, it means the plant's gonna grow. So think about it. If I have a look at it, cell division and cell enlargement means that that makes the plant grow. That's simple. Okay. Next one, we're going to have a look at apical dormancy. Okay. Now, apical dormancy, there's specific things that happen with apical dormancy. Next one, the formation of adventitious roots in cuttings. What does that mean? Adventitious roots. Remember, you get two types of roots. You get tap roots, which are the ones that go right down to the bottom, and you get adventitious roots or lateral roots that go to the side. Right? And normally, when you do a cutting, in other words, like a rose tree, if you cut a rose at a certain point and you have auxin, it makes them start growing adventitious roots. Okay. Next one, we're going to look at the development of flowers and fruit. Okay. We look at abscisic, let me get this right, abscission of leaves and ripe fruit. Okay, and the last one is tropism in plants. In other words, how a plant moves. And I'm not talking about where you see this plant coming, picking its legs up out of the ground, which are the roots, and running around. I'm not that type of movement. I'm talking about how a plant grows according to light or gravity, right? So light, gravity, or water. Okay, so those are the things that we're having a look at when it comes to auxin. Okay, now let's look. The next hormone that we have is called gibberellin. So we've done auxin, which has got to do with growth, right? Now we're going to have a look at the thing called gibberellin. Okay, gibberellin causes elongation of internodes in stems. Remember, you've got a growing point in a stem, which are your nodes, okay? And between these two nodes, it's called internodes, okay? Now, your nodes, if I had to draw it here, your nodes is where your growth points are, okay? So, say for example, you have your branch there, you'll notice that out of this part here, you will grow a leaf, okay? You will never see a leaf here, ever. This is wrong, okay? You'll never see a leaf over here. You'll only see a leaf at the growing points. And what the internode does, the elongation of the internode, makes the plant grow faster. Okay, next one, the development of flowers. Okay, if gibberellin is present, then you're going to start producing flowers. So that means when it comes to springtime, okay, and you start seeing those little buds for flowers, okay, that's because of gibberellin. Gibberellin's busy doing all of that. Okay, next one, as I said, sprouting of buds. See, the buds make flowers, so the sprouting of buds are the ones where gibberellin is present. The next one, germination of seeds, okay? And then the last one, increases the fruit size. So when there's fruit there, makes the fruit get nice and big. If there's a lot of gibberellin there, what's going to happen? Nice, big fruit. If there's not much, you're going to get these small little fruits. And you can notice them in certain apples and oranges and all of this. You get the big oranges and apples, they had a lot of gibberellin, and very small, very little gibberellin. Right. Now, last one that we're having a look at when it comes to plant hormones, okay, very important, plant hormones, is abscisic acid. Okay, so we've had auxin, gibberellin, and now we've got abscisic acid. Now, abscisic acid is very, very important, okay, because it brings about this word here, dormancy, okay, Dormancy means it makes it go to sleep. You know, like you've heard before where some animals go through this thing called hibernation, right? So they eat for all summer long and then they sleep the whole of winter. Yeah, it's similar to that, okay? But it brings down dormancy. In other words, it stops it from working, okay? So it brings dormancy and inhibits of growth of apical buds, seeds, and roots. It stops them from growing. Now, when do you think you'd use that? Okay, remember, when it comes to winter, it's cold. Okay, and if it's cold, we're not going to make as much food. Okay, so what actually happens is a plant goes, okay, during summer, I photosynthesized and I made a lot of different food. Once I've got that food, what happens? In winter, instead of wasting energy to keep all these cells alive, okay, let me get rid of the leaves. Because if I get rid of leaves, I don't have to feed them or keep them alive. So I get rid of them. But if I get rid of them, I don't have food. So what do? We send abscisic acid right around the whole plant. And what does the plant do? It gives it sleep. So it goes into a hibernation. No growing takes place, nothing. That's all because of abscisic acid. Okay, and then last one, regulate abscission of leaves. In other words, when leaves fall off. Okay, next one, plays a role in the opening and closing mechanism of the stomata and transpiration. Okay, so it doesn't just make things dormant, it also opens and closes the stomata. Remember, your stomata have got its guard cells. Okay, there we go. And it opens and closes this little hole that happens here. It opens and closes them. That's what abscisic acid does. Okay, those are the three plant hormones that we're looking at. Okay, very important. Auxin, growth. Okay, the best way to explain it is auxin has got to do with growth and how a plant grows according to light, according to gravity, and according to water. Okay, that's auxin. Gibberellin, it's all about making things beautiful. So in other words, the plants, the flowers, creating flowers, making big fruit. Okay, all of those things. And then you get abscisic acid that happens opposite to gibberellin. It stops things from happening, stops growth, stops elongation. It makes the plant sleep, goes through dormancies, gets rid of leaves. Okay, so those are the three hormones you need to know for plants. Okay, to me, it's the first time that there's a lot less to know about plants than animals. It's cool, huh? Now, here we go. We're going to start off with a couple of questions because I know we always do questions and we need to know these things. Okay, so here we go. Below, the graph below shows the effects of different concentration of gibberellins on the germination of seeds. Okay, so the germination of seeds. So there we go. Let's have a look what we have here. Percentage, remember, just to go over this, don't forget you need a heading, has to have both dependent and independent variables. You'll see germination in percentage. See percentage, germination, okay? And then in time, and then it says in different concentrations of gibberellin, okay? You've got to have both there in a time period. Now, if you have a look here, it says days, okay? From day 1 to day 15, and we have a look. 1,000 milligrams per liter, 1,500 milligrams per liter, 500 milligrams per liter, and 0 milligrams per liter. Okay, so they're all here, and you will see that I have given a what do you call it? A key. Okay, and the question is, let's see, one possible concentration drawn from the one possible conclusion drawn from the results shown above is that. Okay, so now we need to know, if you look at that, what conclusion can you come up to? You just got to choose A, B, or C, or D. So let's have a look. What is A? Gibberellin concentration has no effect on the germination of seeds. Okay, next one. The highest percentage of seed germination occurs at gibberellin's concentration of 1,500 milligrams per liter. The highest percentage of seed germination occurs at gibberellin's concentration of 1,000 milligrams per liter. And the lowest percentage of seed germination occurs at gibberellin's concentration of 1,000 milligrams per liter. So in other words, what they're trying to say is, if you have a look, germination, we're having a look at which is the highest percentage, okay? And we need to see which one is going to be the best. And this one, I'm going to give you a minute to do because I know you can. So it's a minute, and as you're doing the minutes, you will notice I'm going to scroll up and down like this. So make sure that you concentrate on the question and read carefully. Okay? 1 minute. Are you ready? Off you go. So, hope you got that right and it wasn't too confusing as I was moving it up and down, and you got it all, so you could read it quite carefully. Now, if I have a look at it, so I'm just going to turn the page and we're going to have a look, because it's exactly the same question, okay? Draw from the results shown above, gibberellin concentration has no effect, okay? It's exactly the same question. Now, if you have a look, to me, which one is going to be the best? The first one, gibberellin concentration has no effect on the germination of seeds. Of course it does. Look how the germination of seeds has increased. So yes, it has. Okay, so we definitely know A is not there. If we have a look at B, the highest percentage of seed germination occurred at a gibberellin concentration of 1,500. 1,500 is the dot. So there it goes. One beats it. Okay. Look at number C, so it's definitely not that one. If we look at number C, the highest percentage of seed germination occurs at the concentration of 1,000 milligrams. Let's see, 1,000 milligrams is a straight line, so that's definitely right. So it's definitely that one so far. And yes, you can see the answers there. The last one, the lowest percentage of seed germination occurs at 1,000 milligrams. It's not. We've just proven it's the highest. So, of course, it's going to be C, and C's your answer. I'm sure you all got that. That was very quick. Okay, now, another question, okay? A gardener, try this one out. A gardener removes the apical bud from a rose bush in her garden regularly. Why do they do that? As a result, the rose bush will. So if you notice that when we cut roses, we cut the apical bud off, okay? Why does she do that? And explain why. There's 4 different answers. You need to choose one. Produces more lateral branches, okay? Grows taller, remains the same size, produces fewer flowers. I'm going to give you a quick minute, and I need you to understand. Instead of just choosing one, tell me why. Okay, 1 minute. Are you ready? Off you go. I know you sitting there saying, please, man, this is basic stuff. Okay? It is. Let's have a look. Same question, and there's your answer. It's definitely going to be A. Okay, let's have a look why. You had to choose one. First one, A, of course, was produce more lateral buds. B grows taller. It can't grow taller. Remember, if that is your top, and then you've got your apical stem there, your apical bud, right? If I cut this off, okay, there's going to be no more growing point there. So what's going to happen? The growing point over here will start growing out, and here as well. And at the end, I'm going to cut that one off, and I'm going to cut that one off, and what's going to happen? We're going to start growing out there. Okay, can you understand what's happening? It's going to produce more branches, so it definitely can't get taller. Next one remains the same size. It won't. It's going to get bigger and fatter. And then lastly, produces fewer flowers. Rubbish. Okay, remember, you need to cut all it does, we cut it, we prune it down, so we don't waste all of those places. We can use more energy on the flowers to look pretty, so it'll produce a lot more flowers. Right, so hopefully you enjoyed that part. Okay, those were just plants, and when we get back, we're going to start on with the human's response to the environment. So I'll see you straight after this break. Cheers. Welcome back. Now, we did the plants, right, and all the hormones that go to the plant and how the plant responds to the environment. Okay, so we did the auxin, gibberellin, and abscisic acid. Okay, because hormones are getting used in plants. When we come to humans, we've got those funny things called nerves, okay? And in animals, we have nerves. So we have a nervous system, which is divided into two sections. Remember, it's got your what is it called? Your peripheral nervous system, okay? Right, your peripheral nervous system, which is everything that's not part of your brain and spinal cord, okay? And then you got your central nervous system, which is your brain and spine. Okay, so those are the two. So what we're going to do today, I'm going to go over the certain things, like, for example, the senses. So I want to have a look at certain things. Like, I'm going to start off with the ear. I'm going to start off with the difficult ones, and I'm going to help you with the difficult ones because the easy ones I know you can get. Okay, so this is a revision session. So the more I give you the difficult things, the easier it becomes. Okay, so if we have a look down here, we're going to start off with the human ear. Okay, and you need to know these things very carefully, and you need to know them very well and how each thing work. Okay, we're going to start off quickly with a pinna, which is this guy over here. Okay, very important to have a look at your pinna. It's, if you have a look at me, okay, face to face, we're standing in each other, and these things are sticking out like this. You'll notice if somebody's talking to you and you do this, you can hear more. Okay, that's exactly what the ear does. Okay, now start off, if you look at my ear here, and I start off here, and I move into my ear and I come across, you'll notice that sound comes along and gets trapped here by these things here. Notice how they curve around. And if you follow them, okay, so it comes in, follows all the way round and into your ear over here. So that's shaped for a reason. It's got those little cups things on the sides for a reason. It catches sound and it moves it with all its little way that it's designed to go into the hole that we need, okay? That's why we have these, to catch the sound, okay? So that is your pinna, okay? From the pinna, we're having a look at a tube, okay? Now, so far, your pinna and the first tube, okay, is called your external ear, okay? It consists of the pinna, which is the anatomy, large, fleshy, extended part of the ear, function, collects sound, and channels it into the ear, okay? Now we have the external auditory canal. External auditory canal, all it does is it creates a channel or a pipe for it to move through. And that pipe is stopped right over here. So here it is. Let's say that is the, let's do that again. There it is, okay? And it's stopped here by this membrane, okay? So the sound's going to come in and it's going to be stopped by a membrane, okay? That membrane, when we were younger, was known as the eardrum. Okay, it's known as the eardrum, and that eardrum is there for a reason. Okay, we want to take these sound waves and we want to change them into a different type of movement so that we can change it into energy that we can hear. Okay, so here we go. We have your eardrum over there, as you can see it. Okay, the eardrum, what it does is it vibrates. Okay, and when it moves slowly, it gives those deep bass sounds. When it moves fast, it's the high-pitched sounds, okay? Now, from that eardrum, there it is, it's over there, okay? And that eardrum is joined to 3 ear ossicles, okay? So it's bones. And they're known as the hammer, anvil, and stirrup, okay? Hammer, anvil, and stirrup, okay? Now those, you have your first bone, it goes up like this, which is joined to your eardrum, okay? Then you have the next bone that goes like that, that's joined over there, and then you get the last bone that moves across over there. Those 3 bones, as the sound comes and moves this tympanic membrane, okay, it moves the 3 ossicles. And as it moves the 3 ossicles, now the ossicles are using movement to produce just movement, should I say. As it's producing the movement, we're going to start using that movement. Okay, it ends off at a specific place. Okay, so let's look. We've looked at those. I'm forgetting about the semicircular canals. We're not worried about that. We're worried about the, here we go, the cochlea. Okay, now the cochlea is very important because that's where we get our sound from. Okay, and that sound is very, very, very important. Okay, the cochlea looks like a snail. So it comes, if I have a look at this, it does this. It looks at something like that. It comes across like that again. And I'm trying to get it so we don't go over. That's what it looks like. And the sound travels up here. Oh, wait, let me just do this. And let me change the pen so you can see what's going on. I'm going to use pink. The sound travels in here, runs up this, all the way to there. Okay, once it reaches there, it goes back down again and then out a different hole. Okay, so there's 2 pipes. Okay, you've got the scale of vestibuli and the scale of tympani. Okay, now what happens is there's 2 pipes. So these pipes here that I'm showing you there look like this. They run one next to each other. Okay, so we're going to go that way. Oh, let me do this, it makes it easier. Okay, I'm going to do this. There's the pipes, okay, and you'll notice that the sound travels that way in one, gets to the top, turns around and comes back down the other. Okay, that's how it works. Okay, and what happens is these little bones, these 3 ossicles that you get to this cochlea, or the snail type thing, and it comes to a thing called an oval window. Okay, so you're going to end off with this last little bone, this one here, which is the smallest bone, and it looks roundabout, it's got a foot plate. It does that. It's a foot plate. Okay, and that foot plate, let's say that my arm now is the snail or the cochlea. There's a little hole. Okay, it's an oval hole, and this last little bone, the stapes, okay, it fits in here. There's a little membrane there, and what it does is as it moves, it pushes into this hole. So it pushes into the hole all the time. And inside this cochlea is a liquid. And as I push here, what am I doing to the liquid? I'm pushing the liquid. And the liquid sends that vibration all the way to the top and all the way back down. But as it comes all the way back down, you're going to end off with another little hole called the round window. If that round window wasn't there, what would happen is this would just move like this. It wouldn't be able to go in. I need it to go in because I've got to displace the fluid. So as I push it in here, it pushes out here. But that goes push in all the way to the top, come back down to the bottom, and here it pushes out. So it has to have the round and the oval window. Right. Remember, I'm just doing this fastly. It's a quick recap. Okay. Then you've got this thing called your eustachian tube. Okay. Eustachian tube, if you notice, is over here. It joins, right, the middle ear. It joins the middle ear with, for example, the nose, the throat, and the nose, the throat, and the ear together. Okay. Now that's very important because remember, let's say I've got my other pipe here. Remember, this is the tympanic membrane. Okay. If this is blocked, okay, and I'm pushing this membrane, instead of being flat like that, I start pushing it like that. There's a lot of pressure here. I need to get rid of that pressure. And what I do is I yawn, and then that tympanic membrane shoots back. So this eustachian tube is very important because it keeps the pressure on this side of the eardrum the same as this side on the eardrum. So if there's more pressure on the outside of the ear to the inside of the ear, what happens? We decrease the pressure through the eustachian tube. If there's more pressure on the inside than in the outside, we decrease the pressure. So either way you're looking at it, that one there is either to increase the pressure or to decrease the pressure of the inner ear. That's what the eustachian tube is for. Cool, huh? Now, that is the quick rundown of the ear. Sorry, I did forget the semicircular canals, and you know what that is. That's for movement, where this, you know when you feel unbalanced? Okay, that's for balance. It's got little hair-like structures inside the semicircular canals, and those pick up moving like this, and moving like this. So watch again. Head left and right, okay, up and down, and twisting. Right, so those are the three. So you'll notice the one goes that way, one goes that way, and one goes that way. It's 3D. So one goes up, one goes left, and one goes that way to make it up and down, left and right. Right, those are how the semicircular canals. There's liquid in there, and as the liquids move, it messes around with hair. Okay, so let's have a look. Quick questions for you, because we need to know our questions. First one, the diagram below represents a part of the human ear. Okay, identify, I'm just going to do that one. Identify A, B, and E. So let's see, I'm going to do this. Let's see, A, B, and E. A, B, and E, that's at the bottom of the ear. So I will do that, and I'm going to give you a minute. The only thing is, let's see, explain the results if part A is blocked. And then last one, I'm going to give you more than a minute for this one. Explain what happens if part A is blocked, and then describe how part D, the semicircular canals, play a role in maintaining balance when the body changes speed and direction. So listen carefully. Okay, here we go again. Part A is this piece here. Okay, and then lastly, the semicircular canals, which is part D, which is this piece here. Okay, how does that play a role? So I'm going to give you 3 minutes. Reason for it? I'm going to first do that, A, B, and C, identify A, B, and C. Okay, after a while, I'm just going to move it down so you can see question 2 and 3. So I'm going to give you 3 minutes to do that. Are you ready? Off you go. let's have a look at the answers. Now, quickly, if we have a look at it, please identify B, A, and E. It's very simple. E is the eustachian, your A is the eustachian tube. B is the round window. Wait, where's B? B is the round window. Remember I said to you, there's the oval window here, so I'm going to erase this quickly. You'll see there's the oval window, there's the round window, and then E is your cochlea. Those are very, very simple. Next question was, explain the result if part A is blocked. Part A is the eustachian tube, you've labeled it, okay? Air will not be taken in. So in other words, air will not be taken in, releasing to equalize pressure on both sides of the tympanic membrane. The tympanic membrane and ossicles will not vibrate freely. This may lead to the tympanic membrane bursting and therefore leading to loss of hearing, should I say, deafness or pain. Remember, if this thing is blocked over here, if this side is blocked, okay, if that side is blocked, then this tympanic membrane over here is not going to be able to move. Okay, and then the last question was, let's see if it's on the next page. The last question, describe how part D, the semicircular canal, plays a role in the maintaining of balance when the body changes speed and direction. Remember the three semicircular canals, and it is changing speed and direction of movement causes the endolymph to move in part D. The cristae, which are little hair-like structures found in the ampulle, are stimulated and convert the stimuli into impulses, which is transmitted via the auditory nerve to the cerebellum, okay, from which impulses are transmitted via a motor neuron to the skeletal muscles to restore balance in the body. Remember, sends it to the cerebellum. The cerebellum says, oh, your head is tilted like this. We need to fix it. The muscles, all your body must turn or move something to change it. And that is why when people spin you around, okay, all that endolymph is turning in those semicircular canals, and the body say, oh, wait, hold on, a little bit left, oh, wait, a little bit right, and it keeps moving you because it can't make up its mind on how things are moving. So spinning gets your endolymph to move like that. Cool, eh? Okay, you need a break. We've done quite a bit, and I think you've worked very hard, so I'll see you straight after the break. Cheers. Well, welcome back. What we've done is we've done plants, right? And then I thought to myself, you know, when it comes to response to the environment, the best way to start this off is what you're doing at the moment, okay? You are listening to me. And the last thing is you're seeing me, okay? So we've done the listening and we've worked out how we listen. Now we need to have a look at how you are seeing me. So let's have a look at the board. We have the eye, okay? And the eye consists of certain parts. If we have a look at it, we've got the cornea. The cornea is a cover that covers around the eye to protect it. Then you've got the iris. Okay, and if we have a look here, here is the iris. Okay, the iris is the colored part of your eye. Okay, you've got the lens, which is a thick part here, which is very important. Excuse me, very important. You get a boldy edge over here, if you see me coloring it in, if it will color in the same time that I'm talking, okay, that bulgy part is called aqueous humor. It's filled with a liquid, okay. You have your suspensory ligaments, which hold your ligaments in place, okay. You have your ciliary muscles, which control what happens when it comes to your what do you call it, your lens, okay. You get your ciliary body. Okay, the back of the eye, you get your sclera, which is the back part of the eye. You get your choroid, which is where you're going to find your, it's a thin layer around the eye over here. You get your retina, which is the yellow part, which consists of your rods and cones. You get your arteries and veins, because remember your eye still needs oxygen and food in that. You're going to get your vitreous humor, okay, that's found inside the eye. Remember, it's still a liquid found inside your eye, okay. You're going to get your fovea, which is a very, very important place, because your fovea, what it does, that is called the spot that you want, okay. Any image that you want done has got to be found at the fovea. That is the be-all and end-all spot. Okay, if you want something in focus and to see clearly, you want it to be focused on that fovea spot over there. Okay, then you get your optical nerve, which you can see comes out here. If anything is in focus over there, you are not going to see it. Another name for that specific spot there is your blind spot. Okay, remember, you're looking at rods and cones. Cones are colors, rods are black and white. Okay, those are the main things on the eye. Okay, please remember, if I go over it quickly again, okay, the ciliary muscles, or the iris, should I say, not the ciliary muscles, the iris makes your pupil go big and small. And the pupil is that black thing. Sorry, I forgot to tell you pupil over here. Your pupil is a little black hole. It is a hole. Don't think that it's an object, it's a hole. Okay, so that hole, to get the hole bigger and smaller, we have to move the iris. Okay, now light doesn't make the iris open and close. Okay, it's a big mistake. Yes, if there's a light, the iris, if there's too much light, the iris closes. If there's too little light, the iris goes big. That's why at night, when it's dark, you get this big pupil, and during the day, you get a smaller pupil. Okay, but it's not the light that does it. I mean, yes, it does, but it's not. Okay, the iris controls how much light to go through. So picture this. Okay, light goes through. Light comes through here, hits the back of the back on the retina. Okay, the rods and cones. The rods and cones sends a signal down back to the brain, down the optical nerve to the brain. The brain says, "Hold on." That is very, very bright. That's not going to do it for me. It sends an impulse back to the iris and says, "Iris, listen, but please close that pupil a bit so that we can actually see the image properly." Then the iris closes. Okay, so it's not the iris itself that is sensitive to light. It's how quick the brain responds to how quick. And that is why you see bright light and then you close your eyes and then you come back and look. Okay, it's why you seem to be blinded in the beginning. It's because the rods and cones are picking up all this light. Okay, it's the brain that tells the iris to close. It's not the light. Okay, if you have a look at your lens, okay, the thicker the lens, the more light bends. Okay, if light bends, it means it's going to put into focus here. So if light comes in, what happens is light actually comes in at this angle. Okay, it first gets to the aqueous humor, and at the aqueous humor it bends a bit. Okay, so you'll see it bends a bit. Then it goes through the pupil. Okay, when it gets to the what do you call that thing? The lens. Okay, let me move this up a bit. When it gets to the lens, it starts bending even further, and then it's got to hit the back of the eye in that motion there. Okay, so you're going to have little man standing up there. Okay, and over here you're going to have little man standing upside down over there. Okay, that's how the brain picks it up. Okay, so it's got to swip over, and if it's in focus over there, it's got to be in focus over the fovea, the important spot. Okay, these are all the quick ways of looking at it. Okay, now straight into questions. Let's see how good you actually are. Okay, because I love doing this. Each diagram below represents part of the human eye. Okay, diagram 1 and diagram 2. So it's part of the human eye. The moment to me, it looks the same. Almost. Okay, then it says, give the letter and name of the part that contracts to change the shape of the lens, A. B, controls the amount of light that enters the eye. Okay, and C is protected by the conjunctiva. There we go, the conjunctiva. Those are the questions for you. And guys, these are papers. When I get these questions, you can have a look. I'm taking them from a life science paper, November 2015. Okay, so these are questions, not me making it up. These are questions that you guys are going to get. This is the type of questions you are going to get. Okay, so I'm going to give you, let's see, give the letter and the name. So I'll give you 2 minutes to quickly do that. No, actually, in fact, you need to speed this up. I'm going to give you 1 minute to give me the answers for that. Remember, the letter and the name of each of those parts. Are you ready? Off you go. Don't tell me that was too quick, because I know that you've done it quick enough. Okay, so first of all, if we have a look at it, give the letter and the name for each of the parts. First one, contracts to change the shape of the iris. Okay, contract. So if you have a look, it's your ciliary muscles. It's A. Okay, it's not your ciliary ligaments. Okay, it is the muscles that contract. Okay, so it is A and the ciliary muscles. Next one, controls the amount of light that enters the eye. Okay, it is your iris. So it is your C. Iris, which is C. Okay, come on, C. And then the last one, it's protected by the why can I not say this? Conjunctiva, okay, and that is your cornea. If we have a look at it, where is your cornea? Can you see? It is D. Okay, so there are your answers that should have been done nice and quick. Right, okay, next question. Remember, I love these questions. Same pictures, study the diagram 1 and 2. What process is responsible for the change in shape of part E? Okay, so which is part E? What is responsible for the change of that? And which diagram 1 or 2 represent the state of the eye when a person is reading a book? Ooh, that's quite a big one. Okay, this one, I'm going to give you 1 minute as well. Concentrate and let's get it right. Are you ready? Off you go. Let's have a look at these answers. Remember, it's very quick. The first one was, what is the process in study diagram 1 and diagram 2? What process is responsible for the change in shape of part E? Remember, part E is this piece here, which is your lens, and it's called accommodation. Okay. The accommodation. What actually happens is we need to bend light. If something's out of focus, okay, you've all seen as grade 12s, you should know about your microscope. And you know that you move that microscope in and up and down, right? You move the stage up and down. What happens is it gets it into focus. Now, our eye can't do that. So instead of doing that, it changes the thickness and how thin the lens is, okay? So that is very important. Okay, now the last one, the next question was, which diagram 1 or 2 represents the stage of your eye when a person is reading a book, nice and thick, okay, which means it is diagram 2. The thicker it is, the more it bends, because if I had a look at something that thick and something that thick, what happens is you got more chance to bend. Okay, more chance to bend. This should start bending sooner. Okay, I don't know why I keep doing that. Okay, I'm going back. Let's do that. I'm so used to that's nice and thick and a little bit thinner. There we go. Okay, as it comes in, it bends more. As it comes in, it bends more. This one, it's only going to bend very little. Okay, you'll see how it bends only a little. Okay, the thinner it is, the more it bends. The thicker it is, the more it bends. The thinner it is, the less it bends. And when you're reading, it's right close. It's got to take all of that and bend it as quick as possible. Cool. Okay, now One thing I want to have a look at it, your reflex arc. Just want to go over this to make sure that you know this, okay? Your reflex arc, you've got certain ones you've got to have a look at, okay? You've got your receptors in the skin, okay? For example, it picks up a scent, okay? It comes all the way down, okay, up to the reflex arc, okay? What is that? That's your sensory neuron. Remember, your sensory neuron, when it gets into the reflex arc itself, remember the reflex arc is found inside your spinal cord, okay? You get an interneuron, in other words, it's between neurons, okay? So you get your interneuron, it changes from a sensory neuron to an interneuron, it moves back through a motor neuron, and it tells the muscles to do something. So you get the afferent and the efferent. Afferent arteriole, I'm going afferent arteriole, see how I'm thinking. The efferent neuron, and you get the effector, okay, and the efferent neuron. Afferent, it was affected, the neuron is affected, it goes all the way into the spinal cord, and it comes back through the efferent, okay, which means it's going to do something, and you're going to get the affected area and the recepted area. Okay, for example, if I prick myself with a pin, okay, it goes all the way up my arm to my spinal cord, then it comes back to my arm and it gives the whole arm, something. Once it's gone to here, it sends two messages, one back to the arm, one to the brain. So the brain goes, hold on, I know why you did that. Remember, it doesn't know, the spinal cord can't tell you that it was your finger that was pricked. It doesn't know. It knows pain. So what it does is it moves the whole hand away, not just the finger, okay? Where if it was the brain would tell only the finger to move. I thought I'd finish off with that, okay? So hopefully you got it all right. And I know you're studying nice and hard. Reflex arcs isn't too bad, but hopefully I helped you out this time. And I will see you soon. Cheers.